So thank you, Alain, for two things. First, for organizing this meeting. I'm learning a lot. Second, for inviting me, but it's natural because Alain is a friend. And third, you will forgive me because this will not be developmental biology, but it may have some echo for some of you. So this is just a scheme that summarizes the machine that goes after what Erin has presented. Because after the mRNA synthesis of protein, there is still a lot of job. And here you see a lot of processes which are summarized in that little scheme. And in fact, what is interesting is that all of them have been, are, or will be regulated sometime during the life of the synapse. So there is a lot of room for regulations, including its protein synthesis and all these processes that goes after. And in the talk here, I will focalize, I will put the focus on one synapse, which is sometimes neglected, which are inhibitory synapses, although I don't wish you a life without inhibition, but for some of you, sometimes it may help. Anyway, what I'm going to focus on is on the receptor for inhibition, which are accumulated in front of synapses. And this has been quite a long story, which started in the early 80s when Heinrich Betz purified the glycine receptor, which actually was the first receptor to be purified from the central nervous system, and to be purified and identified and localized. And we found in 85 that the receptor is here present, accumulated in front of the presynaptic active zone where the transmitter is released, and it forms microdomains which are stabilized by a relatively complex but so simple compared to excitatory synapses, a molecular network which is called a postsynaptic scaffold. And that postsynaptic scaffold ensures that the receptor is indeed accumulated. And that was where we were until we did the first experiment and with Daniel Choquet and confirmed with Maxime Dahan in 83 that things are not simple. And what you see here in the background is a culture of spinal cord neurons. Active synapses are stained in red by FM dye. And if we do some undersampling and label receptor with quantum dot, this is real-time imaging. You see that everything is moving. The receptors are moving in and out synapses. If you look, for example, here, it gets in, out, and so on. So the question is, how is it possible that we have something which is relatively fixed, which are the postsynaptic density with the receptor being accumulated there? So it's a long story, and to make a long story short, I will do a lot of shortcut today. What we did was to follow the receptor. So this is a dendrite. This is a knock-in mouse where the inhibitory synapses are labeled, and you see the receptor can be either outside of synapses. This is a trace from a single receptor, traveling, staying for a while inside synapses, getting out, getting in, and so on. So the receptor can be in or out. And if you look as a function of time, you've got a sort of spectrum of position, the receptor being either out or in. And it looks very much like a patch clamp recording. I will come back to this issue a little bit later. You can derive dwell time, the time it will stay in or out. And if you look at large fields and look at the probability of detection of a quantum dot, which is an undersampling situation within synapses as a function of time, you see a noise. This noise reflects the fact that molecules are coming in and out. And if this is an ergodic system, it also reflects something related to the density of molecules. So this is why, despite the fact that molecules are moving all the time, when you do a snapshot with a fixation to look at the localization of molecules, you see accumulation of receptors because they have a certain probability to be in, and this probability is something related to density. Now I will make a little bit of what we're going to speak about today, which is now we are looking, this is a synapse, this is a noise of molecule coming in and out. And indeed, people are very much interested to what are the consequences for the brain. So if you go up here and look at the most emergent situation, you have a situation where it's very difficult to understand. And what I'm going to speak about today is about the back regulation that may act here on the synapse to tune to tune the amount of receptors and the molecular dynamic. Actually, the synapse is at the size of about 100 to 400 nanometer for the postsynaptic differentiation, at which some basic molecular phenomena, such as Brownian diffusion, are well described by statistical physics, but they are regulated by integrated biological processes which are generated at higher scale of organization. This is actually where biology and physics meet without falling in what my friend Alain called the physicalist illusion, which is a way to criticize this kind of approach. But I understand some points. I think it's wrong also in some other related to that subject. But anyway, so what I'm going to show you here today, these are some examples of this kind of regulation that we've been working on the past. And we found that activity, phosphorylation, I will show you some example, agonists and antagonists, or allosteric modulators, or the GABA-B, or integrins and extracellular matrix, or molecules coming from the glial system, including microglial system, I will show you an example in few slides from now, are able to regulate this process here. And this is just a scheme that resumes various pathways that can be used, such as activity and others, regulating diffusion dynamic, and therefore diffusion dynamic and trapping at synapses, and therefore the amount of receptor, and consequently the strength of synaptic transmission. So just to continue with this, I remove a few slides on microglial cell because Sonia did present you that extremely well. So what we did was to modify the activity of microglia. So you all know now what is microglia, how important they are in the brain. They feel not only physiological purpose, but also very important, make a very important role in the pathology. And what we did was to either to activate this microglial system using LPS, or blocking the microglial system using minocycline. And what we found, which is shown here, is that at synapses which release both glycine and GABA, so you've got release of both glycine and GABA, you've got both glycine and GABA receptor at the postsynaptic side. And what you find is that when you activate, for example, the microglial system, you, for example, change the steady state of receptor at synapses, which goes with a change, as you will see, on the number of receptors for glycine receptor, but not for GABA receptor. So you've got a system where you've got a co-release, you've got co-localization of receptors, you activate the microglia, and you regulate one receptor and not the other. Now, how to analyze that? So to do that, we used something that has been derived, that has been used by David Colloon in the mid-80s to study the channel kinetic, studying noise. So we have a noise. Here is a noise, and we can analyze this noise using a Fourier analysis and then a Lorentzian distribution. And from that, it's possible to look at the transition of the receptor coming in and out. And from that, you can derive these kind of curves. And from these curves, you see first that it is not modified for the GABA receptor, as it is shown here, while it is strongly modified for the glycine receptor. And in fact, this allows to access the dwell time of receptor at synapses and find that there are indeed some regulation that take place here for the glycine receptor, but not for the GABA receptor. So this is really a new way to analyze molecular dynamics that I'm presenting you here, where you see, for example, that the total amount of receptor at synapses are composed by two groups of receptors, some which are very stable, and other which are swapping in and out. And it's possible to dissect out, for example, in this case, after LPS treatment, you see that there is a global decrease in the amount of glycine receptor at synapses. The stable fraction here is reduced, and as you can see here, the transition frequency is also slightly reduced. So there are a different regulation taking place for the receptor being superbound, as I call it here, the R star, and the one which are swapping. So this it's not just for the physics, it also has some relevance because, as you know, there are some cases where you've got activation of the microglial cell and disinhibition. This is during neuropathic pain in the spinal cord. So it's a quite interesting system where you see that there is now a differential regulation for glycine and GABA receptor. I'm just going to show you the next slide, and this will be a resume of many papers where there is also a tuning of the GABA receptor by activity. And in this case, we decrease pharmacologically the activity by using drugs, and you see that the confinement increase. This is because you see that this curve, I don't enter into the detail of that, but the more bent it is, the less the molecule can escape from the synapse, the more it is trapped. So this captures the receptor, and the consequence is that the size of the minis are increased. And when you increase the activity, you see exactly the opposite effect, which is a release of inhibition and the receptor which diffuse away by means of lateral diffusion. So this process, okay, so far so good, but this is not a mechanism. This is really a description of what is going on. And we wanted to go deeper. So this is just a resume of that situation relating receptor diffusion and activity. So you see here that it is a calcium-dependent mechanism, but most mechanisms are calcium-dependent. But you see something very important here, which is that this phenomenon depends on the receptor type. You see that, for example, the effect on mobility is not the same for AMPA receptor, GABA receptor, and glycine receptor. And even more, we had the paper accepted this week showing, for example, that the various subunits of the GABA receptor plays a role in this very fine regulation. Now, these regulations also involve phosphorylation at different levels, involving CAM kinase 2, calcineurin, or PKC, or PTK. And I'm going to come back to these issues. This is just to mention that. You have to keep that in a corner of your memory somewhere. And we come back to this point of regulation by phosphorylation. Now, if we really want to understand what's going on, and this is really the purpose of what I'm going to speak about today, which is to try to understand. This is a scheme that I proposed almost 8 years ago, where the receptor is extrasynaptic. It is diffusing. It can be bound or unbound to scaffolding molecule outside of the synapse. It may diffuse in, free, or bound. And then it will be within the synapse. This is a color bar here. And there it can be either stabilized or mobile. This is the R star and the RZ that I have shown you just before, where one complement of the receptor are very much stabilized, while the other one is less stabilized. So if we want to understand, we have to take into account the structure of the system. So this is a synapse seen from above. What you see here are some transmembrane elements such as adhesion molecules or neuroligin or anything like that. What you see here, the donut in green are receptors, are scaffolding molecules which are under, and the small green dots here are the cytoskeleton, the meshwork of molecular interaction below the plasma membrane. And if you see that when a receptor enters and diffuses by chance, it may bind to receptor, see an obstacle here, bind again, and then get out. So you have a very complex dynamic that takes place here that reflects all the chemical interaction that may be seen here at the level of the synapse. And here, let's put it in another way. This is time. This is the energy. You see that the receptor which is diffusing may see small barrier of energy due to the cytoskeleton, higher one seen due to obstacle like this one. And when it sees a donut here, it's like a potential well. It is trapped for a while, then it gets out. But what is also interesting is that when a molecule is trapped here, it becomes an obstacle for the other. Therefore, this is a very nonlinear system. Now, we want to understand what's going on there. And for that, we need a large number of techniques. I'm very much technology-oriented, as some of you know. So single particle tracking, FRAP, Fred Flim, fluorescence correlation spectroscopy, electron microscopy, and so on, plus some modeling to access the microscopic and dynamic structure of the molecular interaction that take place here in this synaptic area. So the first technique we've been using, and this is a technique we use a lot these days, is a palm microscopy. So you are all aware of what is palm, but I just remind you this in few slides. So the principle here is we label, for example, in this case, the Geoffrin, the scaffolding molecule of the inhibitory synapses, and you see what we see with the classical fluorescence microscope are dots like this one. It's not possible to have a better resolution. This is about 500 nanometer in diameter. But we label it with a molecule here which can be photoactivated, change the color. And if we do it with a relatively low intensity, which is a molecule coming one by one, it's possible to determine very precisely where they are. And then it's possible to map the appearance of the molecule. And here you see two things. I will come back to that, but intuitively, you understand that it's possible to count something, for example, the number of occurrence, whatever it means, and it's also, you can see that there are differences in density, in local density of molecule. And I will come back to this point also in a minute from now. So from that, what people do most of the time is to generate images, and I just show you two with images because I like them. This is a same molecule, not this one, but the same one as this one, with a classical fluorescence. You will not be able to do anything better even with a great confocal microscope. But with palm microscope, you can see the structure which is below. So you see that there is a lot of structure which is below, complex organization. And this is a way to do super resolution with little money, because this is much, much cheaper than any state microscope. And it has also an advantage. It's easy to use in culture, very difficult to use in slices, but it's doable. But it has an advantage. It is that the Z resolution is much better than with a STED microscope. And this is an example. I just wanted to show you that because of the beauty of the image. This is a field. You see synapses. And in red are the scaffolding molecule. In blue are the receptor on top of it, because we label the receptor on the extracellular side. So you see that there is we have a very nice 3D possibility of 3D reconstruction. Now, if we go back to science, if we look at this scaffolding, this postsynaptic density, they are moving all the time. And these movements are regulated by the cytoskeleton. This is just an example of the movement by pointing the center of mass of this postsynaptic density as a function of time. And we have shown, I'm not showing you all the data, that this is regulated also by activity. This is regulated by calcium. The microtubules are involved. Actin is involved. And all what can be play a role here is indeed present. Now, if you see this movement, this means that within the structure, you have a certain amount of molecular rearrangement. And this is what we wanted to see. So on the next slide, I'll show you an example of postsynaptic density. These are 7 minutes recordings in the loop. And what you see here are the modification of the microstructure of the postsynaptic density as a function of time. And this is the first time that we could access this kind of what I call K-palm, for kinematic palm, where we can access these kind of changes in real time. So you see here that, in fact, these structures, which are supposed to be stable, the half-life of the molecules in these structures are in the range of about 30 minutes when we do FRAP experiments. So this means that this is a rearrangement of the molecule within the structure. And this rearrangement can be followed by various approaches. And this is the case here, where you see the molecules detection as a function of time. And this is within one frame the detection that appeared, and in blue and in red the one that disappeared. But in fact, they disappear from here because they may have moved, and because it is a stochastic activation, it may have bleached also during the time. But you can reconstruct this on the average and show globally what are the level of change of molecules, which in fact reflect molecular displacement. So what can we do with this molecular displacement? What does it mean? So to approach this issue, we did this kinematic palm. So this is an example of a postsynaptic differentiation in front of a single bouton, which have, in fact, probably multiple active zones. I don't enter into details. And for that, we define two parameters. One parameter, which is related to the compaction, which is this broadening parameter, which tells you something related to the density of molecule. And there is another parameter, which is a planar flow, which tells you how far we go from something which is like a round. I mean, it goes elliptical, and you can have indentation. And now when you look how one evolved, one versus another, you see that you can follow here the broadening, the compaction, the planar flow, which is the ellipsoid shape, and you see that there is a kind of ballistic curve here, and this curve, in fact, tells you something about the energy which is involved to maintain the structure. It is an effective energy. I'll come to this point in a minute. Now, what is interesting is that it can be regulated, and this is, for example, examples with BAPTA, buffering the calcium. This is a control, and this is by increasing activity. And you see that there are different positions here in this graph telling you that there is a way now to quantify this molecular dynamic and show that the molecular dynamic will vary depending on the treatment. Now, if we assume that we are dealing with a 2D fluid and look at what energy does that represent. So this, in fact, this scheme here, this is the same as that one. And we look at the iso energy, which is in kBt, as you can see here. And it tells you that if it was a fluid, if we want to maintain molecules close enough, you need to spend some energy. So, and if you let the fluid go, it will diffuse away. But if you will reframe this diffusion, for example, by molecular interactions, then you can find some iso energy here, which represent the way you escape from, you are less and less circular or less and less elliptical. To make it more understandable, it's shown here. So something that will be represented as a postsynaptic density. The planar flow will look at the elongation, for example, in this case. So in one axis, you will have the compression axis, and in the other one, you will have the elongation axis. So this is equivalent to a pressure, to a strength that you impose to the structure, or you can have it all around, and from that, you derive this kind of curves. Now, if you look now at the position of the different treatment, you see that they may vary a lot. For example, when you buffer the calcium, so this is the mean, and you see here the coefficient of the standard deviation, and you see that you may have a very a large variation in the compaction of the molecule and the freedom of movement, telling you that there are differences in the energy involved. So what I'm showing you on the next slide is, you see it's from yesterday, 5/25, so it's very recent data. But you see here the ballistic curve that links the planar flow and the broadening as a function of time, and you see two things which are extremely interesting. First, you've got a control situation. These are two examples, control situation, and you see that you are not on the same iso energy. But what you can see here is that it is changing its shape within a similar iso energy, telling us, in fact, that what we are dealing, so these are the energy as a function of time. You see that it fluctuates much more when you buffer the calcium. These are the different levels of energy, but it tells you that we are dealing with a system where, and I did the drawing this morning, so it doesn't look very nice, but this is a postsynaptic differentiation seen from above. It has a kind of hexagonal matrix which has been postulated. There are two kinds of binding, high affinity, which are in red, and low affinity, which are in yellow. And in fact, the yellow can be unbound, but the red cannot be. And we deal finally with a system which is just like being linked by springs, because the molecule can be deformed, and this deformation accounts for this change within the maintenance of a certain level of effective energy, which maintains the stability of the structure. Then, how many molecules are involved to do that? So recently, in collaboration with Christian Specht, which actually did most of the work in the lab, we developed a technique where we activate the fluorochrome, and we look at the bleach of each of these activation, and we count the number of steps for the decrease of fluorescence. And this number of steps, in fact, represent a number of molecules. So you count all the steps that you have here, and at the end, we end up with about 220 scaffolding molecules in a postsynaptic differentiation. And we developed also another technique to do that, which is, in fact, to look just at the fluorescence drop, and at the end of the fluorescence drop, you can see here these kind of events, which represent the fluorescence which is emitted by a single fluorochrome, and from that you can calculate the total number of fluorochrome, and we end up with the same number. But there is something very amusing in that with these data here, with this fluorochrome, you can use the pointing accuracy and build up an image from what is almost completely bleached. And this is an example of what we get. So this can be done with any fluorochrome. This is the fluorescence of a postsynaptic differentiation, and this is built. This is a super resolution image built with these signals which are at the end of the fluorescence, and I call it napalm for non-activated photoactivated light microscopy. So it's a way to get super resolution at even a cheaper price. So in the last, in the few last slides, I'm come back to this issue of this issue of the binding here and trapping of the receptor. So is it possible? What we wanted to do is to access the binding energy for the receptor at the synapse, the distribution of dwell time, and of dwell time, because the distribution of dwell time is 1 over the k off, and the time to reach a synapse, because this is 1 over k on globally. So to do this, we use a high density labeling of single molecules with 2 different approaches, either using palm microscopy or using the U-pen techniques, which have been developed by Janon in the lab of Daniel Choquet. And from that, you can access high density, map at high density of diffusion with short traces, and build up a map at the surface of the cell where you can look at the density of point, and from that you can reconstruct trajectories using what is called posterior probabilities, and from these posterior probabilities see how far you are from what you expect from a pure Brownian diffusion. And from that you can access the energy which is involved in the trapping. So this is a postsynaptic density. What you see here are the iso energy, and the dots are there are 2 different clusters in this PSD, and the dots are the position of the molecules as a function, integrated over time. And from that you can deduce here in KBT, you can deduce the depth of this potential well. So can this be regulated? So is it something genuine? So a few years ago, in collaboration with the laboratory of Schindelin, in collaboration with Günter Schwarz, which we did a lot of ITC together with him, what Günter found was what defined what the pocket where the receptor bound. And where the receptor bind here, we identified already at that time this serine 403 as probably being important in the binding of the receptor to the scaffolding molecule. This is a place where 2 molecules of geoffrin dimerize. Now we are much, much farther than that, but these are old data, but they really make the point. And what we identified this serine 403, and by so we did design a new tool to study diffusion. So we put the loop here within under a transmembrane domain, and this transmembrane domain is free to diffuse, so it acts as a random scanner in the surface of the neuron. And this is an example where it runs at the surface of the neuron, and you will see it diffusing, and will enter here into the postsynaptic differentiation, have a different behavior. So from that, we could look at the properties of diffusion, and only 1 mutation here, this mutation of that serine, was able to reduce the binding of the receptor and therefore increase the diffusion. All the other one had no effect here on diffusion. The gray is a control situation. So what is interesting is that when we went back to try to look at the depth of the potential well, what we found was that that depth here was really depending upon that phosphorylation. You see here that there is a decrease in the trapping energy when we mutate this serine 403 to add here a pseudophosphorylation. This is shown here in a more simple way, where you see that there is a drop. This drop, in fact, lead to a decrease. This mutation, decrease to drop in the depths here of the potential. So we can indeed also have access to the first passage time, and the first passage time is 1 over k on, and the dwell time, which is 1 over k off. So it's possible now, with this technique, using the dynamic properties, to access the true chemistry of molecular interactions in living systems. So this is the real last slide here. So now we have access to a certain number of data. The nanoscopic organization, molecular counting, binding energies, fluctuation in these energies indeed, dwell time, which is 1 over k off, and first passage time, which is 1 over k on. And we have in parallel developed the theoretical tools which allows us to really make a story out of all these numbers, which take care both of the molecular dynamic at a single molecule level, take care also of the geometry of the neuron. This is a very important parameter, and we are working a lot now on that, and also take into account the chemical potential and the diffusion reaction. So this is a picture I like because these molecules here are very nice. This is a bird fly. These are starlings in the sky in Italy. And you see that there is a quite complex behavior which is being studied now by people doing statistical physics that in fact resemble very much the molecular movement here. And actually what is interesting is that the algorithm and the approach that we use is quite comparable. So this is just the conclusion here. To acknowledge the people which have been involved in that work. So the people which have been involved today in the work I presented to you today are mainly Christian Specht, Pamela Rodriguez, which is a postdoc, a very gifted postdoc, Leandro, which is a physicist, Yasmin, which is a PhD student in the lab. And this was made possible because we had a lot of collaboration with physicists, either experimental physicists or theoretical physicists, like this one. We did also the work in collaboration with the group of Günther Schwarz for ITC and measuring energies for the binding. So thank you very much.